Hello everyone, in this video I'm going to be talking about something called mitosis, which is the scientific name for cell division, or specifically cell division in body cells. And by body cells we mean not sperm and egg, every other cell except sperm and egg. So we're going to talk about why mitosis is important, why you need cell division. We're going to outline the actual steps involved in it. So let's think why mitosis is so important. In fact, we'll outline some of the reasons at the top here. Cell division is required ultimately for growth. If cells can constantly divide, then we could form tissues, so an organism itself can grow. But also, in that, if any tissue is damaged, by having cell division, we can repair that tissue. So having cellular division allows for repair. So growth and repair is typically, when you first are introduced to cell division, the reasons given for mitosis, for growth and repair of our bodies. But there is another important reason why mitosis is important. It allows for something called cell differentiation. Now, there is something called the unicellular organism. Those are organisms that are or exist as one single cell. They have to adapt to any environment that they are exposed to. If you can divide, if you can become more than one cell, we refer to you as multicellular. So by undergoing mitosis or cell division, you can have multiple cells, and by having multiple cells, you can have cells that differentiate or become specialized to become different things. So for example, we have cells that become rods, cones, sperm, egg, heart, liver, kidney, muscle. We have all these different cell types, and that's only possible, that differentiation is only possible because of mitosis, because we've become multicellular, we've developed more than just one cell. So there are three key reasons, growth, repair and cell differentiation, as to why we need mitosis. Now we're going to look at the actual steps involved. So I've said in a previous video about the cell cycle that the cycle starts with a period called interphase. Now, interphase is a period of growth for the cell, so it develops all the new organelles, it synthesizes all of the structures you found typically within the cell, so the nucleus, the membrane, the endoplasmic reticulum. You get synthesis of everything in the cell, all the proteins needed. But within that interphase, we'll put the name of the phase here, so within that period of interphase, you have a stage called the S phase where the DNA is copied or replicated. And that's going to be our starting point in this video. So in the cell cycle video I talk about interphase in more depth, but we're going to start from sort of the end of interphase, if you like. So we're going to imagine we've got our cell here. We have, I'm just going to draw the nuclear membrane, very simplistic pictures here. Now, in a typical body cell, we have 23 pairs of chromosomes, so 46 individual ones. Now for this video, I'm not drawing all 46. Instead, I'm just going to draw a single pair, because even with that single pair, we can demonstrate what happens in mitosis. You just have to kind of visualize and build up imagining there are 46 of these. So in interphase, let's take one pair. We're going to draw one chromosome here, and we'll draw in red. We'll have another in blue. Now, as I said, you get DNA replication. So we can't have one cell dividing to become 2, then becoming 4, 8, 16, without copying the DNA. Otherwise the amount of DNA, the number of chromosomes would halve. So instead we're going to copy them. So we'll have a second red one there, and we'll have a copy of this blue one here. Now, just to be clear, in each pair of chromosomes. So in the 23 pairs, one comes from mother, so we call it the maternal chromosome, and one comes from father, the paternal chromosome. So in this, we'll say the maternal chromosome is the red one, and we'll say the paternal one is the blue. So in interphase, we've had this growth of the cell and replication of the DNA, and that's why we can see a replicated maternal and paternal chromosome here. Then we move into a distinct phase called prophase. Now, in prophase, those chromosomes shorten and they condense. When they condense, they shorten, they become a bit more visible. So we have this nuclear membrane, and we can start to see these chromosomes now as two actual separate strands almost. Looks a bit like this. And I'm going to explain what I'm drawing in just a moment. So we have this phase called prophase, and in prophase, the chromosomes condense and shorten. They get fatter, and we can see them as two distinct strands, if you like. Our original strand, so our original chromosome from mum and dad, the original red and blue one, and their copy. That's why they appear as two. Now this black dot that I've drawn in the middle, this is called the centromere. That's where the two chromosomes, the original and its copy, are joined to. So they're joined at the point called the centromere. And just one thing to note, we called originally, if we just take, imagine, we'll draw it in fact over here. If we take the blue paternal chromosome and its replicated copy and put the centromere in, we initially called this strand here the chromosome, but when they're as a pair, we call each one of those strands a sister chromatid. That's really important. So now we're going to say that each blue strand, if you like, and each red strand is a sister chromatid. So in prophase, we get condensing of the chromosomes so they become visible as two strands. This nuclear envelope will disintegrate, and we get two protein structures forming called centrioles. And they form and move to the edges of a cell like this in prophase. So we'll just shrink this down just to create a little bit more room. We'll talk about the next step. After prophase, we have a period called metaphase. Now, meta refers to middle. So in metaphase, the chromosomes line up. So each pair of sister chromatids line up in the middle of the cell in what's called the equator. That nuclear envelope, if you remember, has disintegrated. So now we have a red there. We have the blue paternal chromosome here and its copy. And we have at the side our centriole, our proteins here. Now what happens in metaphase, as we said, is that these chromosomes, if you like, these or sister chromatids from each pair, line up in the middle of the cell called the equator. And these centrioles produce what are called spindle fibres. And these protein spindle fibres attach on to copies of those chromatids. So this one on the left here that I'm drawing will have a spindle fibre that will attach on the left side to one of the red strands, one of the copies there, and it will have a spindle fibre that attaches to one of the blue strands, one of the chromatids in that pair. This centriole on the right hand side will grab if you like, the other two. And then those spindle fibres contract, they pull inwards towards themselves. So if you imagine these spindle fibres pulling in this way, and these ones pull that way, you would pull those sister chromatids apart, and then that leads into a phase called anaphase. Now, anaphase basically is just a phase when we see those chromatids being pulled by those spindle fibres. Looks a little bit like this. If we just draw a very rough sketch. So in anaphase, we've had those chromosomes and their replicates, forming essentially chromatid copies, line up in the middle of the cell, and then they're pulled to opposite ends. So these two pull that way, these two get pulled this way, and that's what we call anaphase. Now, actually, at key stage 3 and 4, the names of the phases aren't required. So where I've written interphase, prophase, metaphase, anaphase, they're not needed until A level or IB. You just need to appreciate what is ultimately happening. So far, all we've had is the DNA copy and replicate, so that it becomes visible. Centrioles form, spindle fibres form from those centrioles, and they pull chromosomes and their copies to opposite poles or ends of the cell. So after anaphase, we have telophase, and in telophase, what we imagine is something that looks a bit like this. So I'll just draw and then I'll explain what we've got here. So in telophase, when the individual chromatids are pulled to opposite ends of the cell will then start to form a nuclear membrane around that DNA. So we can see it almost starts to form the two cells. So we get nuclear envelopes forming around the separated DNA, and then the cells, or the cell rather, starts to split. We see this what's called invagination. It starts to come in, where I'm just drawing this red arrow here and here. And it starts to invaginate to be able to produce two cells. And if you follow that through, if you imagine we get a sort of split here, that's what happens in telophase. That one cell divides into two. Now there is a phase that sometimes books refer to, sometimes they don't, called cytokinesis, and it's the name of the phase when the very last bit of those two cells decide to split. So in telophase we have two cells forming, and at the point where I just draw a little asterisk here on this diagram, at the very last point where those cells start to divide, that we call cytokinesis. So we can see after telophase, again if I shrink this down, just move this up a little bit, after telophase, what we've ended up with are two cells, draw the nuclear envelope around both, and in both of those cells we can see one red and one blue chromosome. So just one pair in each. And that is what we started with. Now, if we look at interphase at the top, we can see that I've drawn the pair of chromosomes, the maternal and paternal, with their copied version. But originally there would have been just two chromosomes in that cell. So we started with two chromosomes, and in both of the cells we've made, down at the bottom here, both those cells, they both have two chromosomes, that single pair. And that is mitosis. That is cell division of body cells. We go from one cell to two. The two cells that we form are identical. So some key things about mitosis. The cells are identical that we form. So these cells are identical. We are essentially going from 1 to 2 cells. And both cells that we form have a full set of DNA. So they're what are called diploid cells. Diploid means have a full set of chromosomes. So mitosis goes from 1 cell to 2. The cells that you form, the daughter cells, they're more specifically called the daughter cells that you create, are identical copies of one another, and they're diploid. They both have a full set of chromosomes. And this video is just to outline the processes of or involved in mitosis. In another video, I'm going to be explaining meiosis, which is when you form sperm and egg, and it's very, very different to what we've drawn here. And typically both are taught together. So once you've revised mitosis, it's worth having a look at the video on meiosis to see the differences between the two processes, because that one is the one where you introduce genetic variation. But this, you're not. You're making identical cells, one cell creating 2 copies, both diploid, for growth, repair, and cell differentiation. Okay, hope all that helps.